Hi, everyone. Thank you for the kind words, and thank you, everyone, for coming tonight. As David mentioned, my name is Cambrian Liu. I am a new research assistant professor at UChicago, and the focus of my research is examining whether stem cells might change how we think about and treat inflammatory bowel disease, or IBD. Next slide, please. First, a little bit about me. I grew up in Baton Rouge, Louisiana, and I've had the great pleasure of living in some of the best food cities in the US. Along the way, I also happened to get some degrees. At Chicago, the focus of my research group is on identifying new strategies to treat patients with IBD. Next slide, please. As many of you are familiar with, IBD affects millions of Americans and millions more globally. Most of our patients are being treated with medications that suppress the immune system. The problem is that many patients don't respond or respond only incompletely to these medications. And so many patients are actually left behind by the current regimen of therapies. As a result, new medical approaches and therapies are desperately needed. And the focus of my research is actually on trying to heal the wounds that this disease causes within the intestines. Studies have shown that healing of these wounds is essential to get optimal therapeutic outcomes in IBD patients. Next slide, please. My lab's approach toward doing this is to leverage the activity and power of stem cells. Stem cells are potent cells that can turn an embryo into an adult and grow all of our organs. In adults, they're responsible for lengthening our hair. And in the GI tract, they are responsible for maintaining our intestinal organs and also for helping them respond to natural insults and injuries. Next slide. In my lab, we can isolate stem cells by the millions from routine medical procedures such as colonoscopies. We can grow them in a dish into these structures we call miniguts, which are small replicas of that patient's intestines. Next slide, please. And my lab focuses on using and studying these stem cells in three major ways. The first is we want to know if we can transplant stem cells and miniguts back into the diseased intestine and help launch a healing process. This would be really important towards giving patients an important new avenue of therapies to treat their disease. Next. We can also use stem cells and miniguts to predict how a patient might respond to certain IBD therapies. We can expose these miniguts to different therapies and ask whether they disintegrate or proliferate. And that can really help doctors and patients decide what therapeutic strategies might be most appropriate for them. Next, please. And finally, we can combine stem cells and imaging to understand what are the deep origins of IBD and what induces the chronicity of this disease. And this will illuminate new ways we can actually target this disease for therapies. Next slide, please. So I want to leave you with tonight is this idea that we're at the cusp of a revolution in terms of how we think and how we might treat IBD. And stem cells are really at the center of this all. Thanks for listening, and thank you for your support.
Cambrian, there were two questions for you. The first one is, are you a DC or a Marvel Comics fan?
I would probably have to lean Marvel, but I'll take anything that.
You had a few DC folks there in your slides, we all noticed. But the second question really was, how close are we to cloning somebody's intestine and growing it in the lab and being able to use it as a treatment or as a replacement for their bowel when it's had problems?
That's a really great question. I think we are quite close. So many of the technologies that we need to be able to do that sort of work now exist, and that's really been the push in our field for the last 10 years, learning how to isolate stem cells from someone's intestine, learning what conditions you can use to optimize the growth of those intestines. And then we now know, at least in some animal models, how to then put the intestines that we've cloned in a dish back into the animal. Now, with that being said, this is a field that is a mix of biology and also a little bit of engineering, a little bit of computer science and statistics as well. As the other presentations have shown, Ashley's and Ben's and Natty's, IBD is a very personal and personalized disease. And one of the exciting things about stem cell work is that you can, in theory, modify those stem cells slightly to confer some therapeutic properties or some resistance to disease. And that's really, I think, more of the longer term goal of the field as well, is to how can you improve stem cells so that they are more resistant to disease and can be more powerful effectors of therapy. And that will take studies from a lot of disciplines to get there, but we are working really, really hard on it. And I think we'll be there sooner than we think.